Recent in vitro studies show apple-derived polyphenols significantly reduce cancer cell viability.
In this video, I'm going to be talking about how researchers have investigated the long-term effects of apple consumption related to cancer markers, as well as immune and inflammatory pathways. And I'll be using a variety of study types from human cohorts and controlled trials, and even to animal and cellular experiments. So apples are rich in phytochemicals such as flavonoids and polyphenols, dietary fiber and antioxidants, and that interact with multiple biological pathways that influence cancer risk and immunity. So studies have linked higher apple consumption with a lower risk of certain cancers, such as lung and colorectal cancers, over many years of follow-up, and especially for women, that apples was one of the few individual fruits associated with lung cancer risk in women. And some cohort data sets suggest inverse associations between flavonoid intake and lung cancer development up to several decades later, implying a long-term relationship between diet and tumor risk. So these studies don't prove causation, but they support a pattern where habitual consumption of apples or flavonoid-rich diets correlates with reduced cancer incidence over long follow-up periods. Okay, so a notable human feeding study had overweight adults contain 3 whole apples per day for 6 weeks, and there were reduced inflammatory markers like C-reactive protein or CRP and interleukin 6 or IL-6 in plasma. And these markers are connected to both cancer risk and immune system status. Now, when IL-6 is too high, what ends up happening is that increases hepcidin. And there is an iron exporter called ferroportin. So if hepcidin, because of IL-6, is reducing ferroportin, then that means that iron can get trapped in the macrophages and leading to plasma blood iron deficiency. So that goes to show that apples actually helps increase iron levels and iron absorption. Because if there is iron deficiency, there could be a growth in hypoxia. Hypoxia is a very strong driver of M2 phenotype macrophages. So basically, this M2 phenotype supports tumor growth and even protects the tumor. Also, iron is critical not only to reduce low oxygen hypoxia states, but also to make sure that a vitamin A is functional, because vitamin A is reliant on iron. If a vitamin A is deficient, then the parietal cells, they cannot secrete intrinsic factor, because vitamin B12 relies on intrinsic factor for vitamin B12 to be functional and absorbed. And without B12, because of no intrinsic factor, then there's low glutathione. So if glutathione is low, then advanced glycation end products can grow. Also, ages weakens the immune system. That there are a lower number of T cells or more untrained naive T cells, because what happens is that advanced glycation end products, they actually glycate the APOB48. As a result of that glycation, there are less antigen presenting cells. If there are low APCs, then they can't train the naive T cells, because that's the job of antigen presenting cells. So they have a CD70, and then the T cell has a CD27, and that's how the APC trains the T cell, or the naive T cell. But of course, with ages, there's less APCs, therefore less training of T cells and a lower number of T cells. So that goes to show that apples are actually great to support your immune system, since apples lowers IL-6 or lowers hepcidin, so that it allows ferroportin to export iron trapped in the macrophages. Okay, so these studies on cancer are short-term mechanistic effects on immune signaling and inflammation, which are key components of cancer progression and immune function. So, suggesting that regular apple consumption may modulate immune pathways even in adults with metabolic risk factors. So, possibly even in people who are already at risk, or even people who already have cancer or who have already been diagnosed with cancer. Okay, in rodent models, um, in rat colon carcinogenesis models, feeding apples for 3 months reduced early cancer markers, uh, or the precursors to colon cancer. Now, this doesn't mean that it translates over to humans, or that it would have the same effect in humans. It's only in rodent models. And other rat studies using apple extracts showed lower tumor incidence and altered expression of genes tied to apoptosis. So that means that in rats, apple extracts uh, decreased tumor cell proliferation and increased programmed cell death through apoptosis to lower the number of tumor cells. So another thing that apples do, and the phytochemicals uh, studied in cell culture, is that there is a modulation in P13K AKT pathways, which are central to cell proliferation and survival in many cancers. Uh, so basically, higher P13K AKT means higher Ki67. Okay, so if Ki67 is too high, that means a higher increased chance of cancer growth, or in a higher number in cancer cells. But if Ki67 is lower, then there are a lower number of cancer cells and slower cancer growth. So the goal is to lower and reduce that Ki67, which is exactly what apples are able to do by lowering the P13K AKT pathway. Okay, so quercetin in apples actually exhibit antioxidant activity, which can lower oxidative DNA damage, which is a factor implicated in cancer initiation. Now, there's a chromatin, and then inside of the chromatin, there is a DNA. And there are genes and transcription factors that interact with the DNA. So if apples are protecting or lowering oxidative DNA damage, then that means that apples could be enhancing or improving and enabling the interaction of genes and transcription factors with the DNA, because many so basically, like, different immune system functions rely on that gene transcription factor interaction with the DNA in order for immune system functions to work. And also, apple extracts enhanced stress resistance and longevity, hinting at effects on cellular defense mechanisms relevant to immunity and aging. Okay, so quick recap. Apples, notably one of the few individual fruits inversely associated with lung cancer risk in women. Apples help reduce C-reactive protein and interleukin 6, or IL-6, therefore not allowing hepcidin to reduce the ferroportin, allowing iron to be released from macrophages to reduce iron deficiency. Apples protect against DNA damage, therefore allowing interaction of genes and transcription factors with the DNA. So that's it for the video. If you want more future videos, then make sure to like, comment, share, and subscribe, and keep it locked in here on my channel, and I'll see you in the next video.